Hello my dear students. Today we are going to cover the topic of cell division. Have you ever thought what is the necessity of cell division? It is actually for the growth or to create next generation. Let's consider single cellular as well as multicellular organisms. In the case of single cellular organisms, this cell division leads to increase in the number of individuals. But in the case of multicellular organisms, it's either increase the number of cells leading to the growth of individuals or it may increase the number of individuals. Let's move into cell division. The entire events of cell division we can together say it as cell cycle, in which one cell divides into two daughter cells. And generally the entire events, all the sequential events, we can classify it as two phases. The first one is interphase and the second one is M phase or meiotic phase. Now we are moving to interphase. Most of the events, that is 95% of the duration of the entire cycle is happening in this interphase. This growth, development, cell functions, everything is happening in this interphase. Due to this, we can again classify this phase into three. That is G1 phase, that is growth 1 phase, synthesis phase where the DNA replication happens, and again growth 2 phase. Next, we are talking about M phase. I've already said 95% of the duration of cell cycle is happening in the interphase, and the remaining 5%, only this 5% is happening in M phase. This is the shortest phase in a cell cycle. Okay, and in this phase we can see two overlapping events, two sequential events. First one is karyokinesis. We have already said that in the interphase DNA has already duplicated. This duplicated DNA will divide into two nuclei. So nucleus is dividing into two. The division of this nucleus is said by karyokinesis. And the second event is cytokinesis. Cyto means cytoplasm. In this period, the cytoplasm is dividing into two, where the complete cell division is happening. Even though cell division happens both in prokaryotes as well as in eukaryotes, both are different types. Here we are going to discuss about cell division in eukaryotes, which happens in two types, that is mitosis and meiosis. Now we are going to talk about the major difference between mitosis and meiosis. In the case of mitosis, this type of cell division is happening only in somatic cells, in which only one round of chromosomal replication, which is followed by one round of cell division. It will result in the production of two identical daughter cells. Remember, identical daughter cells. But in the case of meiosis, this type of cell division takes place in gamete cells. In this, one round of chromosomal replication is followed by two sequential cell division. The thing is, in this, the daughter cells will not be identical. It will be different that of from mother cells. This is due to the genetic recombination happening in the cell cycle. Now we are going into detail about each phase in cell cycle. So the first one is G1 phase or gap phase. We can call it as growth phase also. So this is the most elaborate or most time consuming phase of cell cycle. So in this phase, cell is preparing itself for DNA synthesis, which is happening before the S phase. Okay, so the major events happening in this phase is this pre-RC complex, assembly of pre-RC complex, that is pre-replication complex, centrosome duplication, everything is happening in this G1 phase. All these events are happening in the late G1 phase or just before, just the early S phase. So these major events are triggered by a protein complex, that is cyclic E-CDK complex. CDK means cyclin dependent kinase 2 complex. Now moving to the S phase or synthesis phase. This is the phase in which DNA replication or DNA duplication, the amount of DNA is going to increase in this phase. So in this phase, the centrosome replication has already completed. So the one thing you have to keep it in the mind is that here the DNA amount is increasing, not the chromosome number. So for your clarification, I am attaching one image along with this video. In the first image, in the first figure, you can see that the number of chromosome is 3 and the DNA is 3. Okay, in the second image, the number of chromosome is 3 but the number of DNA is 6. Okay, in the third image, the number of chromosome is 6 as well as the number of DNA is also 6. Already I have mentioned about this centrosome duplication. I'll be giving you much more detail regarding that. In the G1 phase, okay, usually this centrosome is seen nearby the nucleus. One pair of centrosome is seen near the nucleus as like a cross. So in the G1 phase of cell cycle, this centrosome, this pair will get separated. That is centrosome disjunction. Okay, that this is happening in the G1 phase. In the S phase, these two separated centrioles, budding will form. Buds will start forming. Okay, this happens in the S phase. While the cell moving into the G2 phase, these buds will grow into a complete centrosome. So the complete growth of centrosome happen in the G2 phase, and these two centrosome will move to the opposite poles of the cell cycle. Next is G2 phase or growth phase. This is the phase in which the actual growth happens. Means all the organelles, mitochondria, Golgi apparatus, ribosomes, all the other cell organelles are getting duplicated in this phase. This happens between the S phase and M phase. Now we are moving into M phase or meiotic phase. Okay, in this phase we can again classify into 3-4 stages. That first one is prophase, then prometaphase, metaphase, anaphase and telophase. All these can be considered as karyokinesis, where the nucleus is getting divided. All these events are followed by cytokinesis, where the cytoplasm is getting divided into 2. So all these events I have mentioned earlier, that is karyokinesis. Now the nuclear division is over. The thing that is going to happen next is cytokinesis. That is, the cell has to divide into 2. The cytoplasm has to divide into 2. So as a beginning, the cell in the center, a light curve will be forming. This process is called indentation. So gradually this curve, this curve will get into deeper, much more deeper, and gradually it will get separated into 2. So final result is the production of 2 daughter cells. By this, the process of cell division is over. I hope I have covered the overall picture of cell division. Hope all of you understand this. Thank you so much.